So the last section that we have to talk about when we consider the integument are sensory receptors that are present in the different layers of the skin and the hypodermis. And so the skin is protective and thermoregulatory and has lots of other functions, but one of the things that you use your skin for all the time is to gather information about your environment. And so there are several different types of receptors in the skin, and we're going to talk mainly about tactile receptors, which are going to respond to touch and vibration and stretch. And that's going to be in contrast to some other types of receptors that might be chemoreceptors that would respond to chemicals. Although free nerve endings do also respond to other stimuli than touch. All right, so here's the list of the ones we'll talk about from your textbook, and the two that you can see in lab are Pacinian corpuscles and Myersner corpuscles, but we'll talk about the other ones and their function as well. And so what you're looking at in this really cool special stained slide is the epidermis way up here with the underlying dermis, and then the hypodermis, and you can see very large tactile receptors here that lie in the hypodermis. So this is one type of tactile receptor called the Pacinian Corpuscle, which is huge, and it lies in the hypodermis, and it's especially found on, you can find it in a lot of them in fingertips. And so here is a more close-up view of this type of receptor, the Pacinian Corpuscle. And so it has a consistency of modified Schwann cells that are arranged in lamellae, and the nerve afferent ending is going to enter at a pole of the Pacinian corpuscle, still be myelinated, and then you can see where it ends up here at this dot. And so when you get compression of this corpuscle, it's going to stimulate an action potential in this afferent nerve ending, and then the action potential is going to go back towards the spinal cord as a general sensory afferent if it's in the periphery of the body. And Pacinian corpuscles can also be in the wall of organs, so they can carry GVA information, but we're talking about skin, so that's going to be GSA, and it's going to go towards the central nervous system, and it's not going to hand off its signal though until it comes into the spinal cord. Now the cell body of this afferent nerve ending is in the dorsal root ganglion. So if you're trying to put together gross anatomy and histology, these sensory afferent nerve endings are going to have their cell bodies in the dorsal root ganglion. And so these types of receptors specifically are going to be stimulated by touch, sustained touch, and deep touch, and vibration. And so this type of stimulus, like a vibratory stimulus, will fire off the Pacinian corpuscles. The next type of sensory receptor is called a Myersner corpuscle. It's also encapsulated similarly to the Pacinian. It's made up of modified Schwann cells, but in contrast, they're actually organized in a parallel fashion that a lot of histologists talk about them being like a stack of coins, so going back and forth and back and forth. Very specifically, you're going to see these in the dermal papillae. So the dermal papillae sticking up, you can find Myersner corpuscles here, and you'll do more of this in lab. And so the axon is going to actually come into this corpuscle at the base here, and they are going to respond to light touch, and so this is going to be very much more superficial than the Pacinian corpuscles. Other types of sensory receptors you're not going to see in lab are free nerve endings. So here's a special stain here where you can see free nerve endings. They are not encapsulated, and they oftentimes are going to transmit noxious stimuli. So stimuli that are going to transmit pain signals will be picked up by free nerve endings. Another type of sensory receptor is called a Raffinis corpuscle, and this is a very, very thin connective tissue capsule, and there's a little bit of fluid in here, and then you have collagen fibers that actually stick through the capsule, and these collagen fibers get pulled on when the skin gets stretched and torqued. And so the pull on the collagen fibers will fire the afferent nerve endings that are at the end of the corpuscle and will cause a stimulus. Will cause a signal to be sent to the brain. And so these Raffinis corpuscles are going to be at the junction of the dermis and the hypodermis, and they're going to respond to stretching and torque. And so what I try to hook up Raffinis with in my mind so I can remember them is that the Raffinis are the ones that you feel when you put socks on. So when you put socks on at the very beginning, you feel the sock and you feel the compression and it stretching your skin. And so you feel it right away, but then eventually you don't really feel your socks. It sort of decreases its firing and its signal, and then you don't think about it much longer. So I think of Raffinis as Raffinis socks. Okay, and then the next type of sensory receptor is a Kraus end bulb, and these sensory receptors have a thin collagen capsule. It has one sensory fiber that sticks into it, and it's mainly found in certain places of the body, like the skin of the penis and the clitoris, and it is going to respond to low frequency vibration. And in my PowerPoint, there's a little link here if you want to read a little more about how circumcision changes the stimulation of the Kraus end bulbs. All right, and then we already talked about the Merkle Corpuscle, but I wanted to remind you about it here because it is a sensory receptor. And so these were the sensory receptors that had neural crest origin and epithelial origin, and they were used for reading Braille, very, very sensitive sensory receptors that are going to be very superficial at the right below the basement membrane, or right at the basement membrane of the epidermis. Okay, so that's it. We got a few questions to do, and then you are done.